My name is Mayo Faulkner, and I'm a research software engineer at the International Brain Laboratory. And today I'm going to present to you the Brain-wide Map project dataset. This dataset was collected as part of the International Brain Laboratory, which is a global collaboration formed of 10 theory and 12 experimental labs. It was formed in 2017 with the aim of trying to understand decision-making in the mouse brain. One of the platform projects of this collaboration was the Brain-wide Map project, whose aim was to record neural activity at the single cellular resolution throughout the mouse brain during a decision-making task. This project served as both a scientific initiative and also a sociological experiment to see if we could do large-scale open-source research in a collaborative and distributed way. The task that we used during the Brain-wide Map project was a visual decision-making task. A stimulus would appear on a screen in front of the mouse, and they'd have to use their paws in order to move a wheel to bring the stimulus to the center of the screen. If they correctly moved the stimulus to the center of the screen, they received a reward of sugar water. If, however, they moved the stimulus in the wrong direction, off the screen, they received an error tone. In order to initiate a trial, the mice needed to hold the wheel still for a period of between 0.4 and 0.7 seconds, after which they were alerted of the start of a new trial by the simultaneous presentation of a visual stimulus and an audio go cue tone. Mice had 60 seconds in order to make a decision before the trial would time out and they'd receive an error tone. During the task, the contrast of the visual stimulus was varied, from high contrast trials to low contrast trials to zero contrast trials, where no visual stimulus would be shown on the screen. The probability of the visual stimulus appearing on the left or the right of the screen also varied in blocks of trials throughout the task, with the probability varying from 20% to 80% likely to appear on the left. This change of block was not indicated to the mouse in any way, and so in zero contrast trials they needed to use their prior knowledge of the block structure of the task in order to infer which side the stimulus was most likely to be. There are 3 task events that I want to highlight. The first is stimulus onset, so this is the time at which the visual stimulus appears on the screen. The second is first movement time. This is a time at which the mouse initiates a wheel movement in order to move the stimulus beyond threshold. And the third is feedback time, the time at which the mouse either receives a reward or an error tone depending on the outcome of the trial. This diagram here shows the experimental setup that we used as the mouse performed the visual decision-making task. In each session, we recorded from up to 2 neuropixel probes simultaneously, and on this neural data, we performed spike sorting and also extracted a number of different electrophysiology features. We continuously tracked the mouse's wheel movement and also recorded the outcome and timings of all task-related events. We also recorded from 3 cameras, a left, right, and body camera, on which we performed pose estimation in order to track the paw and extract, for example, the pupil diameter. We also extracted the video motion energy in regions of interest on the different cameras. This meant that for each session, we have a rich dataset where we can link the neural activity to a wide range of different behavioral variables. This figure here shows a recording plan that we used during the Brain-wide Map project. In order to obtain uniform coverage, we used a grid-based system where our insertions were separated by 500 micrometers. In each recording session, we targeted locations that shared anatomical connections. And for this reason, in the cerebellum and medulla, we recorded from the right hemisphere instead of the left in order to account for cross-hemispheric connectivity. Each recording site was targeted by at least 2 laboratories, and in each subject we recorded from one repeated site, called the repeated site, and that allowed us to assess the reproducibility of our recordings both across subjects and labs. In some subjects, we also performed bilateral recordings where our insertions were targeted in the same location but on opposite hemispheres in order for us to investigate interhemispheric interactions. In late 2022, we released this dataset to the public, and it currently consists of the following: data from 12 labs in 139 mice, 459 recording sessions, which comprise of 699 probe insertions, and these span a total of 281 brain regions, and we've detected over 620,000 neurons using spike sorting, of which 75,000 have been classified as good. So how can you access this data? The first way is using the pre-prepared data that we've prepared specifically for the Neuromatch Academy course. So this data contains peri-stimulus time histograms of neural activity aligned to different task events. So the advantage of using this data is it's very easy to use and also lightweight, which means it's very quick to download. And using this data, you can very quickly start doing brain-wide map analysis. The disadvantage is that the data is limited in scope. It only contains neural data and task-related data, but it does not, for example, contain any of the real or video data. To use this data, we prepared a Colab tutorial notebook called IBL Brain-wide Map Neuromatch Tutorial. And in this, we show you how to download and load the preprocessed data, to look and explore the content of the data, and then to perform some single cell and population activity level analysis that are aligned to different task events. The other way to access our data is using ONE, and this is an API that we've developed as part of the International Brain Lab that allows you to access our data. The advantage of using this method is that you have access to the full data set, and it's also linked to a metadata database that allows you to flexibly search and download data of interest. The disadvantage is that there's a higher barrier for entry. You need to familiarize yourself with the API first before you can start using it. And also, as the data has not been pre-curated, the data is larger, and therefore download times and space on disk is going to be longer and larger. We've also prepared a second Colab tutorial called IBL ONE tutorial that shows how to use this API. Here we introduce you to the different methods, how to search, load and download data using this API, and we also walk you through examples of how to perform simple analysis on the different type of data sets that are available. So collecting and preparing this Brain-wide Map project was a major undertaking spanning several years and comprising the dedicated efforts of many individuals. And in this slide, I just want to highlight all members of the IBL, both past and present, who have made this data set available. And with that, I'd like to thank you and wish you good luck on your project.